The overall goal of this procedure is to determine the capacity of sera collected from Zika virus-infected rhesus macaques to mediate antibody-dependent enhancement of dengue virus infection in vitro.
This method can help answer key questions related to antibody-dependent enhancement of infection of dengue caused by Zika virus-induced cross-reactive antibodies. The main advantage of this technique is that it is high throughput, simple, and easy to perform. Demonstrating this procedure will be William Valiant, an emerging infectious disease graduate student from my laboratory.
Begin by thawing the serum samples at room temperature. When thawed, transfer 100 microliters of each serum sample to a sterile tube and heat and activate for 30 minutes at 56 degrees Celsius. Next, make 10-fold serial dilutions of the serum sample ranging from 1:1 to 1:1000 in cold RPMI 10. Then transfer 10 microliters of each diluted serum sample to the wells of a sterile 96-well V-bottom plate. Next, thaw dengue 1, 2, 3, and 4 reporter viral particles in a 37 degrees Celsius water bath, ensuring a volume of approximately 170 microliters of each RVP per serum sample. Once thawed, immediately transfer the tubes of RVPs to ice. Then pipette 10 microliters of RVPs into the wells containing serum sample dilutions and to the no serum control wells. Mix thoroughly by pipetting up and down 5 to 10 times. Transfer the 96-well V-bottom plate to an incubator and incubate for an hour at 37 degrees Celsius.